Intestinal amoebae are protozoa that move using pseudopods. Their infective stage is the cyst, which is usually ingested in contaminated food or water. The diagnostic stage may be either the cyst or the trophozoite, often referred to as the trough, found in feces. Let's start with Entamoeba histolytica. This is a pathogenic amoeba. It can cause ulcers in the intestinal tract and can spread to form abscesses in the liver or lungs. The cyst of E. histolytica contains up to 4 nuclei with uniform peripheral chromatin and a central karyosome. It may also contain chromatoidal bars, which are cigar-shaped, and it measures about 10 to 20 micrometers in size. The trophozoite stage has a single nucleus with uniform peripheral chromatin and a central karyosome. A key diagnostic feature is the ingestion of red blood cells inside the troughs. These measure 12 to 60 micrometers and show progressive motility. Next is Entamoeba dispar. This is a non-pathogenic species that is morphologically identical to E. histolytica. The major distinction is that its trophozoites cannot ingest red blood cells. Because the morphology overlaps completely, serological testing is needed to differentiate E. dispar from E. histolytica. Entamoeba coli, which is also non-pathogenic. It is often confused with E. histolytica. Its cyst contains up to 8 nuclei, but the chromatin is irregular and the karyosome is eccentric. The chromatoidal bars, when present, have splintered ends. The cyst size is 10 to 35 micrometers. The trophozoite stage has a single nucleus with irregular peripheral chromatin and an eccentric karyosome. Inside, it may contain ingested bacteria or yeast. The motility is sluggish, and the trough size ranges from 15 to 50 micrometers. So, in summary, remember: E. histolytica, pathogenic, red blood cell ingestion, progressive motility. E. dispar, non-pathogenic twin, no red blood cells ingested. E. coli, non-pathogenic, many nuclei in cyst, sluggish motility. Let's continue with 3 more non-pathogenic intestinal amoeba: Entamoeba hartmanni, Iotamoeba buchli, and Endolimax nana. Entamoeba hartmanni looks identical to Entamoeba histolytica, but it is smaller in size. Its cyst measures less than 10 micrometers, and its trophozoite measures less than 12 micrometers. Importantly, it is non-pathogenic. Iotamoeba buchli is also non-pathogenic. Its cyst stage contains a single nucleus, lacks peripheral chromatin, and has a large blot-like karyosome. A distinguishing feature is the presence of a glycogen vacuole, and the cyst measures about 12 micrometers. The trophozoite also has 1 nucleus without peripheral chromatin and with a blot-like karyosome. Glycogen may be present throughout the cytoplasm. The trophozoite measures around 15 micrometers. Endolimax nana is the smallest intestinal amoeba and is non-pathogenic. Its cyst contains 4 nuclei, lacks peripheral chromatin, and has blot-like karyosomes. A clear halo can be seen around the karyosomes. The cyst size is about 8 micrometers, making it the smallest amoeba. The trophozoite contains 1 nucleus with no peripheral chromatin and a blot-like karyosome surrounded by a halo. It measures about 10 micrometers. In summary, E. hartmanni looks like E. histolytica, but smaller and non-pathogenic. I. buchli has a glycogen vacuole and blot-like karyosome. E. nana is the smallest amoeba with cysts as small as 8 micrometers. Intestinal protozoa can be classified based on their type of motility. The first group uses pseudopods for movement. These are the amoebae. They are further divided based on the presence or absence of peripheral chromatin in the nucleus. With peripheral chromatin, we have Entamoeba histolytica/dispar, Entamoeba hartmanni, and Entamoeba coli. Without peripheral chromatin, we find Endolimax nana, Iotamoeba buchli, and Entamoeba fragilis. Although Entamoeba is technically a flagellate, The second group uses flagella for movement. These include Giardia lamblia, Chilomastix mesnili, and Trichomonas species. The third group uses cilia for motility, and the classic representative is Balantidium coli. So, by remembering the type of motility, pseudopods, flagella, or cilia, you can quickly classify the major intestinal protozoa. Let's compare Entamoeba histolytica with Entamoeba coli. Starting with the trophozoite stage. E. histolytica has a central karyosome and uniform peripheral chromatin. It shows active, progressive motility and may contain ingested red blood cells, which is diagnostic. In contrast, E. coli has an eccentric karyosome and irregular peripheral chromatin. Its motility is sluggish, and it may contain ingested bacteria. Now, looking at the cyst stage. E. histolytica cysts have 1, 2, or 4 nuclei, and the chromatoidal bars, if present, are cigar-shaped. E. coli cysts can have up to 8 nuclei, and the chromatoidal bars have pointed or splintered ends. So, in summary, E. histolytica is defined by red blood cell ingestion, progressive motility, and cigar-shaped chromatoidal bars, while E. coli shows sluggish motility, bacterial ingestion, and pointed-ended chromatoidal bars with many nuclei. Let's review the free-living amoebae that can cause human disease. Naegleria fowleri and Entamoeba species. Naegleria fowleri is a pathogenic amoeba that causes primary amoebic meningoencephalitis, often abbreviated PAM. This infection is usually fatal. Humans are infected when the amoeba enters the nasal passages during swimming or diving in warm freshwater lakes or ponds. It can also come from contaminated tap water, such as with neti pot use. Once inside, the organism invades the central nervous system. The diagnostic stage is the trophozoite, which is demonstrated in cerebrospinal fluid. Entamoeba species usually affect immunocompromised patients. They are associated with keratitis, an infection of the cornea, and with a chronic, slower form of meningoencephalitis. Unlike Naegleria, they are not typically linked to recreational water exposure. Instead, infections are often related to trauma to the eye, such as from contact lens use or injury. In short, Naegleria causes acute, rapidly fatal brain disease from nasal entry, while Entamoeba causes chronic brain disease and keratitis, often related to eye trauma and immune suppression. Flagellates are protozoa whose motility is achieved through flagella. Their infective stage is usually the cyst, which is ingested in contaminated food or water. However, in those species that do not form cysts, the infective stage is the trophozoite. The diagnostic stage is either the cyst or the trophozoite found in feces. Let's start with Entamoeba fragilis. Despite its name, electron microscopy shows that this is actually a flagellate and not an amoeba. It has no cyst stage and is considered pathogenic. Its trophozoite usually has 2 nuclei, with the karyosome composed of 4 to 8 granules. Interestingly, it lacks visible flagella. Next, Giardia lamblia, one of the most important flagellates. This protozoan is pathogenic and causes gastroenteritis. It is the most commonly reported intestinal protozoan in the United States. The cyst has 4 nuclei, retracted flagella, an axostyle, and a parabasal body. It is ovoid in shape and about 12 micrometers in size. The trophozoite shows a characteristic falling leaf motility, has 2 nuclei, an axostyle and parabasal body, 4 pairs of flagella, and a ventral sucker used to attach to the mucosa. It is teardrop-shaped and measures about 12 micrometers. Infections are common in daycare centers, immunocompromised patients, travelers, and hikers exposed to contaminated food or water. Moving on to Chilomastix mesnili, which is non-pathogenic. Its cyst has a single nucleus, is lemon-shaped, and about 10 micrometers in size. The trophozoite has 1 nucleus and a spiral groove, which produces a corkscrew-type motility. Now let's consider Trichomonas species. Trichomonas vaginalis is pathogenic, while other species are generally non-pathogenic. No cyst stage exists. Species are identified by their source. For example, T. vaginalis is found in vaginal or urethral discharges. Trophozoites are teardrop-shaped and have an undulating membrane attached to flagella. The length of this membrane helps with differentiation. In T. vaginalis, the membrane is about half the length of the organism, while in T. hominis, it extends the full length. Now let's briefly look at ciliates. These protozoa move by cilia. Their infective stage is the cyst, acquired by ingestion of contaminated food or water. The diagnostic stage is either cysts or trophozoites in feces. The classic ciliate is Balantidium coli, the largest protozoan known to infect humans. It is pathogenic. Its cyst is round, has 2 nuclei, a double cell wall, and is about 50 micrometers in size. The trophozoite is completely covered with cilia, has 2 nuclei, 1 large kidney bean-shaped macronucleus and 1 small round micronucleus, is oval in shape, and measures about 100 micrometers. In summary, Entamoeba fragilis. No cyst stage, pathogenic, looks like an amoeba, but is a flagellate. Giardia lamblia. Pathogenic, falling leaf motility, ventral sucker common in U.S. Chilomastix mesnili. Non-pathogenic, lemon-shaped cyst, corkscrew motility. Trichomonas. No cyst stage, undulating membrane. T. vaginalis is pathogenic. Balantidium coli. Only important ciliate, very large, pathogenic.